Traditional sequencing takes days and requires massive centralized instrumentation. Nanopore sequencing delivers gigabytes of data in minutes using a device the size of a smartphone. This technology reads DNA by threading single strands through a nanoscale protein pore embedded in an electrically resistant membrane. As the DNA passes through, it temporarily obstructs an ionic current flowing across the membrane. The unique chemical structure of each nucleotide causes a distinct measurable change in the current. This allows for direct, real-time identification of the base sequence without the need for optical detection or chemical termination steps, fundamentally changing how we approach genomics. The core component is the flow cell, containing thousands of synthetic or biological nanopores, typically derived from proteins like MSPA or CSGG. These pores are integrated into a lipid bilayer or a synthetic polymer membrane separating two fluid chambers containing an electrolyte solution. A voltage is applied across this membrane, establishing a steady ionic current, usually potassium chloride. When a DNA molecule enters the pore, it displaces the ions, reducing the current. The diameter of the pore is critical, often engineered to be just wide enough to accommodate a single strand of DNA, approximately 1.5 to 2.5 nanometers. The ionic current reduction is not uniform, it is sequence-specific. Since the pore is narrow, approximately 4 to 6 nucleotides reside within the sensing zone at any given moment. This group of bases, known as the K-mer, dictates the magnitude of the current blockage. For example, a K-mer rich in purines, adenine and guanine, causes a different current drop compared to a pyrimidine-rich K-mer. The system measures the duration and amplitude of these current fluctuations, generating a raw electrical signal called a squiggle. This squiggle is the direct electronic readout of the DNA sequence passing through the pore. Nanopore library preparation is significantly faster than traditional methods, often taking less than 10 minutes. The primary requirement is attaching specialized adapter sequences to both ends of the target DNA fragments. These adapters contain a hairpin loop and a crucial component, the motor protein. The adapter sequences also facilitate the initial capture of the DNA molecule by the pore. The DNA does not need to be amplified via polymerase chain reaction, allowing for direct sequencing of native DNA. This direct reading capability means epigenetic modifications like methylation are preserved and alter the current signal, providing additional biological information.
Uncontrolled translocation of DNA through the pore would be too fast for accurate measurement. This is where the motor protein, typically a helicase or polymerase, becomes essential. The motor protein binds to the adapter and acts as a molecular brake, ratcheting the DNA strand through the pore one base at a time, or in controlled steps of several bases. This controlled movement slows the DNA passage rate from thousands of bases per second down to approximately 450 bases per second. This ensures sufficient dwell time for the electronic measurement system to accurately register the current changes associated with each K-mer, maximizing signal fidelity. The raw electrical data, the squiggles, are digitized and streamed in real time. Basecalling is the computational process that translates these current measurements into the standard adenine, thymine, cytosine, guanine sequence. Early basecalling relied on hidden Markov models, but modern systems utilize deep neural networks and machine learning algorithms. These algorithms are trained on known sequences and their corresponding current signals to accurately predict the K-mer responsible for a specific current level. This computational step is often performed on a dedicated graphics processing unit to keep pace with the high data output rate required for real-time analysis. The primary technical advantage is the ability to generate ultra-long reads, often exceeding 100 kilobases and sometimes reaching megabases. This capability is transformative for resolving complex genomic regions, such as repetitive elements, structural variations, and phasing haplotypes across large distances. Furthermore, the Mion device, powered by a USB connection, enables sequencing outside of centralized laboratories. This facilitates field applications like outbreak surveillance, environmental DNA analysis, and rapid clinical diagnostics at the point of care, democratizing access to genomic information. In health, nanopore sequencing is critical for rapid pathogen identification, allowing clinicians to determine antibiotic resistance profiles within hours instead of days. For cancer research, long reads help characterize complex chromosomal rearrangements and fusion genes that are difficult to map with short-read data. In agriculture, it is used for rapid plant genome assembly and identifying viral infections in crops. Environmental applications include metagenomic sequencing of soil or water samples, providing immediate insight into microbial community structure and biodiversity without extensive sample processing.
Initial single-pass accuracy was lower than short-read technologies, but accuracy has significantly improved through better pore chemistry, enhanced motor proteins, and advanced base-calling algorithms, now often exceeding 99%. For maximum accuracy, consensus sequencing, where the same molecule is read multiple times or multiple molecules are aligned, is employed. Future developments focus on direct RNA sequencing, allowing immediate analysis of the transcriptome without complementary DNA conversion. Additionally, increasing the throughput of high-density flow cells like the Promethean system will continue to drive down sequencing costs and increase data volume.